All right. Hello, everyone, once again, and welcome to the Town Hall of Q2. That means that we're going to update you on all the things that are happening at Arise now that we are halfway in 2024. And of course, many of you may have been to Arise Day, but we're now 3 months ahead. And again, some amazing things have happened. So feel free to ask questions afterwards or share your opinion so that we can only make Arise better. Just as a quick reminder, who are we? So we are a big project team consisting of 4 different institutes: Naturalis Biodiversity Center, the University of Amsterdam, the University of Twente, and of course the Westerdijk Institute. We are divided across 5 different teams. Each of them has their own assignment, but they're highly interconnected, and so they will present end-to-end workflows together. Now, what are we talking about? Just as again, a quick reminder, we are doing everything around species identification because we feel like once we have a feeling how many species are out there, which species are out there, we can actually start answering ecological questions like who's eating who's interacting or dependent on who. We do this basically on 2 different ways. On the one hand, we have the DNA stream, so from DNA or from sample in the field all the way to DNA identification. And on the other hand, we have a lot of different sensors in the field that automatically register the presence of a species. But of course, that also requires a lot of AI with a lot of data management underneath. Now, first, before we go into Arise, I've actually tried to spice up our town halls a bit by also invited keynote speakers. So I'd love to introduce you to Peter van Bracht. Peter van Bracht is one of our biggest ambassadors, I would say, for Arise. He's been exploring the Netherlands, but even Scotland, to find us specimens for our so-called target list. So for those species that we know exist in the Netherlands, but we do not have a specimen nor a barcode. And Peter, I think you've already acquired a ton of, like hundreds of specimens to us, and also even discovered new species for the Netherlands. So yeah, I'd love you to give us your spiel. Can you share your screen, Peter, and unmute?
Okay, and this one and this one. This should be working. And here is the presentation coming up, I presume. This should be in the picture now. Good afternoon, everybody in the Netherlands. I'm not in the Netherlands myself at the moment, but thanks to all these virtual machines, we can do a lot of things from all over the world. Just to get started, I want to take you back to July 12th, 2020. It was sort of like the beginning of the corona pandemic. Diving was luckily still allowed. And I made a dive near Zierikzee, near the bridge crossing the Eastern Scheldt. It's a nice diving spot. There's quite a bit of biodiversity. It's not pristine for sure. There's a lot of invasive exotics there, but still there's also a lot of native species that can be found there. And one of the species that is quite abundant there in spring, early summer is Sertularia cupressina. Sertularia cupressina, it's a hydroid, so it's a member of the Cnidaria. The jellyfish, sea anemones, corals, and hydroids. And this Sertularia cupressina is a favorable food for various species of nudibranchs. And as you can see, here's another picture of Sertularia. This was actually a picture taken in that somewhere around that time. It's loaded with these nudibranchs, and if you're not really sure what you're looking at, there they are. There's quite a lot of them, but they're very cryptic, at least this species. And it's a Dendronotus species. In Dutch, we call them boompjesslakken. Dendronotus species. And I'm going to give you a little bit of a historical update on how the Dendronotus species in the Netherlands evolved in let's say, our scientific knowledge. Here's a close-up, sort of isolated picture that shows you a little bit better how these beautiful creatures look like. You're looking at an animal. Obviously, it's a mollusk without a shell, about maybe 4.5 centimeters long. This is actually quite a large specimen. But let me take you a little bit back in history, even further to 1773, when Leendert Bomme, he was a merchant. He lived in Middelburg. And he was very interested in marine life. Obviously, he wasn't a diver. Obviously, he didn't have a digital underwater camera to record it. But he just went beachcombing, and in the intertidal areas, he was just looking at all sorts of marine animals and also the seaweeds that he found there. And he once in a while he published something about it. And in 1773, he published a paper in which he describes to some extent this Dendronotus species. He didn't give it a scientific name, so it wasn't really a scientific publication. And he called it het hartshorn gelijkgetakte zeeslakje. Just 1 year later, Peter Ascanius, he's a Norwegian guy who was actually a pupil of Linnaeus. He published a scientific paper with a more taxonomic description. He actually called this species then Amphotite frondosus, which the genus name was later changed to Dendronotus because Amphitrite was already taken for a genus of annelids, marine annelids. But so we missed out on having this species scientifically described by a Dutchman, and the Norwegians got away with it. But until 2017, in the Netherlands, for sure in the Netherlands, we only had one species of Dendronotus, and there was some variation. There were some smaller ones, some larger ones, up to about 12 centimeters length, actually. But we always thought it was that it was just one and the same species, Dendronotus frondosus. Until 2017, there was a paper published by Korshunova and others. And they described how Dendronotus frondosus was actually a species complex. And within this paper, they described several new species, and one of them was Dendronotus europaeus, based on DNA data, but also based on anatomical data and ecological data. They described the species, and it wasn't really to our surprise, but for sure it was good evidence that we had plenty of good evidence for it, that this species also occurred already in the Netherlands for long time. So in 2017, we didn't have one Dendronotus boompjeslak. We had two Dendronotus species, both Dendronotus frondosus and Dendronotus europaeus. And here's actually a picture that was taken a little while ago in the Netherlands near Burgsluis in the Eastern Scheldt of this Dendronotus europaeus. And then if you compare this one with the original picture of Bomme, then most likely actually Bomme already described a species that was only scientifically described in 2017. So interesting finding for sure. But we started at the 12th of July 2020, and these are 3 specimens of the smaller species. So I just assumed this was Dendronotus frondosus near the bridge crossing the Eastern Scheldt. And 3 of them were sent to Naturalis. From 2 of them, we got a nice CO1 barcode. And if we compared the barcode to the DNA databases like BOLT and GenBank, now this might not be very clear. Hopefully you can read it on the screen. I can read it properly, so it should work, I guess. Here on the top you can actually see, I don't know if you can see my cursor. Elaine, do you see my cursor in the picture? Ah, that's brilliant. These are some older specimen of Dendronotus frondosus. So that's the, let's say, the original described species. Some of them already within the collection of Naturalis. here are the CO1 barcode homology data, and they all fit nicely within, let's say, 98.5 to 100% homology. But it turned out that these two new species, and remember this is 2020, the two specimens that were sequenced from that I collected near the bridge, they don't match at all with this population. They actually only share a 88% homology with these initially sequenced Dendronotus frondosus specimen. But they do match with a few others that were already in the databases. There's several. There's one which was identified as Dendronotus frondosus. There's one identified as Year Yargle. That's another species. That's another Dendronotus frondosus. So presumably within Europe, some specimens were already collected of this species, but misidentified and definitely not belonging to the original. This is actually here. This is the reference of a neotype described Dendronotus frondosus right here. So this is definitely another species. And just in the end of, I think it was October, if I'm correct, October 2023, another paper, again by Koshnikova, described a new species of, several new species actually, of Dendronotus. And long and behold, if you look at our data, they match very, very nicely with this new Dendronotus kitlei, described and published in Diversity in 2023. So although we thought that there was so much research already done within Europe about this genus, and we thought, well, there's not much going to be found anymore, especially a lot of DNA research had been done already. So it was quite unexpected that we found another Dendronotus species, this new one, also in the Netherlands. So we decided to propose at least the trivial name onverwachte boompjeslak, the unexpected Dendronotus. I think that's quite an appropriate name for this new species. And now, if you look back in our archives of pictures, slides, and digital pictures, it actually turned out that as far as the small Dendronotus specimens that have been observed in the Netherlands and photographed, most of them, definitely, as far as I can judge by now, most of them are actually belonging to this species. So Dendronotus frondosus, the originally described species, is not all that common. Europaeus and kitlei are much more common in the Netherlands. Now, this is only just one example of what actually ARISE is doing. We are not only logging, let's say, DNA codes for all these known species in the Netherlands, but initially I called it like bycatch, but it's I think that's an understatement. It's really the spin-off, very valuable spin-off of this sort of research and this whole project, that by just trying to get barcodes of species which we think are relatively common, we find quite a bit of species which are actually new to science or are not described yet. And this was one from the original. Well, there were, like, at this time I had about 540 specimen delivered to Leiden. But within these 540 specimen, there were actually many more species that didn't match, or specimen that didn't match with the existing barcodes of the species of which we thought they were. So this is a feline, also a mollusk, which is definitely genetically very divergent of the Feline quadripartita, which is the only known feline from the Netherlands so far. There's a Flabeligera species. It's an annelid, which also doesn't meet, which is not homologous at CO1 level to Flabeligera affinis, which we already thought it was. And actually, this is just a small collection of specimen that I collected within these initially 540. By now, I've delivered over 1,000 specimen already to the RISE project. But there's at least 11 or 13 putatively new marine species that are new to science, or at least new to the Netherlands. So the spin-off of this DNA work is tremendous, and for me, for sure, it's extremely exciting. So I would like to finish with not showing only like these possibilities for the marine species, but obviously, and most obviously, this will also be true for and will also happen to all these other species from the other ecotypes like terrestrial and freshwater, etc. And also well, I mean within any animal or plant group, I'm sure we're going to find a lot of new species for the not only for the Netherlands but also for science. And this is not only happening here. I already knew that this was similar results were achieved, for example, in Norway when they did a similar project like ERISE, but then only for nudibranchs. And there they had like just over 100 species, but they came up with 3 or 4 different new nudibranch species. So just to finish up, well, I truly hope you enjoyed the presentation, and for sure keep up the good work. Send in as many samples as possible, because the more CO1 data we get, the more exciting new species probably will be found. And on behalf of Blauwtipje.nl, that's the organization that Victor Heike and I established. It's a database for Dutch nudibranchs, a database and a website. So also on behalf of Victor, greetings from Karma on the island of Saaremaa in Estonia, because that's where I'm on holidays nowadays. So I'll start collecting again in August, Elaine. Thank you very much. If there's questions, please
And
ask.
thanks so much, Peter. And whoever wants to reach out to Peter directly, just send us an email. So we will connect you. No, fantastic. You're such an ambassador for Arise. So thank you so much also for really displaying from your perspective how much Arise can contribute, not just for just building reference databases, but also your research questions and how that actually expands beyond. Fantastic. So I'm gonna go back to the rest of the presentation. All right, and here we go. And we're going to switch a bit. We're going to go back into the specimens and DNA barcodes again after we have seen some of the work from sensor to AI species identification. So I have to do the honors today because Daniel Kisling, who is running the MDS team, the monitoring demonstration sites team, can't make it, which is the first part, of course, of this presentation. But you see that there's a whole end-to-end stream which actually connects the monitoring demonstration sites team to the AI DSI team and the BioCloud team and vice versa. So what you see here is just a quick overview to once again remind everyone how many different types of cameras we've tested for Arise in the field. And by now it's over 8 cameras. Some of them are more prototypes, others are well established, but weren't necessarily tested in the field yet in the Netherlands. And for all of these, different data is gathered for digital species identification. So you see a lot of camera type sensors, but you also see acoustic sensors, and the radar has also been operational, on which actually Judy Chamous has recently published also a paper. So what's the real purpose of the MDS team? It's to test and deploy multiple sensors for automated species recognition or identification. And so also to develop scientific use cases. What kind of research questions can we actually answer? And demonstrate also biodiversity and the importance of what we do to the general public. We have 3 main locations. Each are chosen for their own different circumstances, but also for their different purposes. So we are at Oostvaardersplassen, which is more of a grassland and marsh area. It's run by Staatsbosbeheer. We are in Artes, which is one of the zoos in the Netherlands, and so a way more city-like environment. And then we have the Amsterdamse waterleiding Duinen, which is owned by Waternet, and it's so obviously a dune area. But on top of that, we have many different deployments all across the Netherlands for various purposes to test out different sensors or help actually already starting those research questions. So they're called our test sites. So let's go into those three main sites and what the purpose has been so far. So for Artis, like I said, it's a city environment. There we have various responsibilities, but one of the major objectives was actually to show the importance of biodiversity and the use of sensor technology to the general public. So a lot of things also happened there or are still happening around communication. There's a radar, of course, but there's several sensors that the public can view, and there's information plaques next to them. There's explanatory signs all across the park. Various articles have been published, and also presentations have been given. And so all to basically get the general public involved with our work. Now, the Amsterdamse waterleidingduinen, so the dune area, is a completely different site, not just for the habitat, of course, but also because we're working directly with one of the stakeholders that have real questions about how to manage their area. And here we're talking actually about large data because we're also putting whole networks of sensors out there. So it's not just testing a single camera, but actually looking into how can we answer a particular question. In this case, they were very much interested in three of their main species that drive the ecosystem. So on the one hand, you have the rabbit, you have the fox, and you have the fallow deer. And so questions are, for instance, how does the relative abundance of key mammal species change across space and time? And do they differ in, for instance, the fallow deer, exclosures compared to grazed areas? So also here, experimental setups have been made to see how much of that changes the biodiversity. But also questions, how do population trends derived from traditional monitoring methods? So for instance, transect monitoring or manual observations compared to changes in relative abundance derived from camera trapping. So obviously both methods have their own pros and cons. And now it's also time to actually make sense. So what are the real things that you observe when you are doing those traditional transect observations versus what the camera actually captured, and can you then start making sense of what's actually out there? So this is all about consolidation. This site has been running for a while and also really serves as an example for our stakeholders. So Waternet, for instance, also adopted some of that technology and know-how that the MDS team has evolved to other sites. Now, last but not least is the Oostvaardersplassen, which is a nature reserve. And here, one of the key species are the geese, or groups of species, I should say. And here, big questions such as, well, can we monitor what the geese are doing? And can we see then how that affects the grazing as well? So here you can also see that different sensors have been tested, different setups, to really get a sense of, okay, how can we capture the geese through these sensors? Because some of the sensors or some of the angles weren't perfect, but now we've learned how to actually do that. And this is then also immediately connected to some of the AI that needed to be developed. So I would say the monitoring demonstration sites team of Arise has actually already fulfilled all of its purposes that was designed by Arise, but they're continuing, of course. So they're continuing the PR development in Artis. There's going to be a walking route and another magazine article. The geese monitoring at the Oostvaardersplassen is maintained, basically exploring the AI output now that also the AI algorithm is working. They're definitely also going to maintain the large-scale monitoring at the Amsterdamse waterleidingduinen and actually involve student projects to do more and more ecological question research. And we're working together with EU scale biodiversity monitoring projects. So we're actually scaling up from just doing the Netherlands to European project levels. One of those is TEDMON, where there's a lot of acoustic monitoring happening. The goal is to develop a transnational biodiversity monitoring with autonomous acoustic sensors across a large latitudinal range in Europe. They're connected to WorkMax 3. And so basically what it means that across the sites that we have, we are now also deploying these bug devices to do acoustic monitoring. We're also working with another biodiversity pilot, which is called ABMS. This is also a pilot for transnational automated monitoring, but in this case for birds, bats, and nocturnal insects through sound and image recognition. And so also in this case, it means that we are deploying additional sensors that are basically monitoring those type of species. So we're scaling out the number of sensors that we have in our sites so that we are actually working together across Europe, which is, I think, quite amazing. And another one is the collaboration with Mambo, also a European project. The goal is to develop, test, and implement tools for monitoring conservation status and ecological requirements of species and habitats for which knowledge gaps still exist. Again, quite a mouthful. And what you see here is the Amy. Again, this is for nocturnal moths. But there's also more prototype type of sensors being tested. And in this case, it's for sedum roots, where they want to track insects. Again, these are deployed in our test sites. So what is this team doing? Well, it's larger scale, so not necessarily huge changes month by month. I know that all the sensors are currently in the field, so hooray for Rotem. I know she's in this call, and if you have any questions afterwards, feel free to ask them to her. So we're analyzing all of those scientific use cases. The Diopsis use case is still very important, and can we analyze then also there the traditional sampling methods with traps versus Diopsis cameras? The new sensors are being deployed also for the European projects. And of course, we're going to facilitate further use of automatic wildlife camera networks. And I'm going to hand over to Chantal.
Yes, thanks, Elaine. All right, all that data needs to go somewhere, right? So, Elaine, I think I'm not a co-host, so you need to do the clicking for me. So BioCloud is the brain of Arise, as we say. And what we try to do here is ingest a lot of data from different sources. If you can click, Elaine. Yeah, lots of different data sources, like the different sensors that Elaine just showed that the MDS team has been put out in the field, but also data related to the DNA side of things, as Peter showed. But we've got a BioCloud and we've got a core sequence cloud, so I'll come back to the DNA side later. And then once we've processed all that data, we want to make sure that people can use it. So in the Arise platform that is under development, we of course want to make sure that people can explore the data, but also use different services like the algorithm services to analyze that data, or look up your samples and your DNA codes in the DNA dashboard. So earlier this year, this was kind of where we were standing. We finally had a stable BioCloud production environment in which we developed the first pipeline for all the Diopsis insect camera images with over 26 million images. That really challenged the Arise DSI team to display that all in the portal. But this is a pretty stable environment that now we kind of can update this daily. So the team's been working really hard in the back end to make this more scalable, to add all the other data sources that we would like to add. And one of the things that we're working on is a API endpoint so that external sources can actually push media to us. So in the first instance, we're going to use this for the MDS team to push the data from all the other sensors, like the wildlife cameras and the site recorders, to the BioCloud so that it can be delivered to the DSI team for analysis with the algorithms, but we'll also open this up in the future for external users to deliver data to us. Nothing public here yet. Some other work in progress. If we, building one pipeline for one use case is one thing, but we're now really making this a robust and scalable data management platform. So we're working on a lot of integration testing, so we can test the whole pipeline, but also all the components separately. Doing a lot of data quality checks. So we are kind of the in-between data sources and the data consumers, in this case, the DSI portal. And we want to make sure that data that we ingest from a data source is indeed the same as what you can see in the DSI portal. So there's quite a lot of quality checks, validation, making sure that it's all correct. And of course, these kind of back lane, these kind of checks are also important, are already the baseline for whenever we add other data sources. And then the other main thing, and I'll come back to that later, that we've been working on is the DNA pipelines. And for that, we needed quite a bit of improvements for the code base so that we could actually share the code between BioCloud and Core Sequence Cloud. Then we can go to the next one. One other very important topic for us is access control. So who are our users and how do we know what this user is allowed to see and do? So identity and access management has been on the radar for quite a bit. People are already able to log into the DSI portal with their own account if we've added them. But what we still needed to do was to make sure that we then use authorization to make sure that they can only see their own data. So one of the things that we, for example, oh yeah, can I now? No, yeah, next, Elaine, I saw the. So what we, for example, retrieve from a data source, in this case, Vonabit, is the authorization information. So Vonabit is the provider of the data, and they know who can see what data. And what we've built in between the API from Vonabit and the DSI portal is a system where we can link those Vonabit rights to the information in BioCloud and have an API endpoint that the DSI team can access to easily retrieve that authorization information. I think that was it from the BioCloud perspective, and I'll come back to you later when we talk about Core Sequence Cloud.
Thanks again, Chantal. And I think this little image of this person I want to analyze just my images from my cameras is very valid for your story here. Thanks. All right, Jacob, you're up.
Yeah, good afternoon, everyone. Elaine, did you reshare the presentation or is it still the same one?
If I started Q2, so.
Okay, can you maybe reshare it? I added one slide, which is quite nice.
Awesome.
Just in time delivery is how it's called, right?
That's great, Jacob, for the presentation.
Yeah. It's
Okay,
good to take a small break, right? There's all this information being blasted to everyone.
True.
So
can you still see my screen in presentation mode now?
Yeah, this is great. Thanks. Okay, now we're all refreshed. Let's continue with the sensor to AI story. So we're now in the middle of this nice timeline. So we are working on AI, but also a method to explore your data through the media browser, which is what you see here. The idea is that you actually browse through your data using filters to organize it, structure it. So if we go to the next slide, we see the available filters. So since the last town hall, we've added the bottom one, a deployment, which allows you to filter your data based on deployment information, which means a time period, a location, and the configurations to that. So this is a GIF that we just added and shows you how fast the system currently is. So now you see Jonty browsing the system, zooming out a little bit, going through 25 million media items. This is an infinite scroll. And this is real time, right, in production. Now Jonty is adding labels to some invalid data, and then adding a filter to exclude those images. Yeah, so it's using the label filter, exclude, and then selecting the label he just added. Then it's updated, so now it's this is a form of data cleaning. Then when you're done filtering, you give your data set a name, you store it. And then you want to start an analysis. So you select an algorithm, a data set, you click start, and then this is the analysis job overview where you see the job is now starting for you. So now we can go to the next slide. Will be a little bit of repetition, but this was nice to see how swift it currently is, right? Another thing we've done is improve the support for audio. So now if you have media audio files in the system, it is displayed in gallery view as spectrograms. So if you know how to read spectrograms, kind of gives you a nice view already about your WAV files. Next slide. So what you've seen happening very fast, but now a little bit slower. So to take you through this process. So first you create filters. Based on those filters, you create a data set and give it a name. And then you want to analyze the data set using AI. So we have an algorithm repository. So if you go to the next slide, you see the algorithms we have currently integrated. So this ranges from the Diopsis insect cameras to common wildlife camera algorithms, geese counting, and almost ready is the Aves Echo bird sound classifier. It's ready. We're going to ask Dan Townhall to actually test it on his audio data. So next up, which we're actually starting this week or next week, is the cricket and cycade classifier. So this algorithm is developed by a Capgemini change and can be used to detect crickets and cycades in audio files. Also, we're developing algorithms to detect prey in avian nest boxes, and Voltes Hogeschool is working on flower biodiversity algorithms, which we are planning to integrate as well. Also, in the next couple of months, we want to start integrating with Hugging Face. Some of you may know it, some of you won't know it, but this is a very popular platform nowadays to openly share algorithms. So if we integrate with this, it will actually make life easier for developers to develop algorithms that are compatible with Arise, but at the same time also open up a large range of algorithms that we can use in Arise. So this is the screen where you select an algorithm and a data set. And then if you click Start Analysis, your data is sent to the Snellius supercomputer all automatically. And when it's done, you can download the results. Now, on the next slide, to infinity and beyond, until now, there was a data set size restriction due to Snellius limitations. Chantal is our super user, needed to run millions of the Diopsis images, so it's mostly a pain for Chantal at the moment. Now this restriction has been resolved. So basically a data set could be infinite until we run out of compute credits, of course, because large jobs are now automatically chunked and handled per chunk for you. So we go to the next slide. So first you had a small job. Now we chunked the large job into sets. This all happens in the background. Now you see here that the blue boxes, so these chunks happen sequentially right now. This means that in time. So the next step, the next slide, is horizontal scaling. So we are now chunking these large analysis jobs, but since Snellius is a supercomputer, which means hundreds of compute nodes available to us, we can run all of those jobs at the same time on different compute nodes, which will tremendously speed up the analysis result. So this is for us the next step. All the information about running analysis jobs. I took this screenshot this morning. So these are all Diopsis analysis jobs running. We have refined the information that you see. So now it also shows you that it's copying data because it needs to copy all those files to Snellius before it can start processing them. As that has been refined, it's also more robust at the moment. So when the analysis job is done, you can actually view individual media items to see what the output of the algorithm is. So for images, that is bounding boxes around the detected insects or geese or wildlife images. On the right side, you also see checkboxes for various algorithms. So if you have multiple algorithms, you can actually select the results for each analysis job you have run, which allows you to quickly investigate what the differences are visually. And of course, for each job, you can download the results in JSON and CSV. Now, another thing, so this is a nice GIF that I got from a different project. So we don't do this tracking in the Arise, not this type, but it's a nice animation of what tracking does. So on insect cameras, but also in wildlife cameras, subjects will be visible in multiple images, right? So if you're detecting and counting species, you don't want to recount the same individual. So we use tracking sequences to deduplicate detections in time series data. So Diopsis also has a tracking module, which first needed to be run separately. If you go one slide further, Elaine, at the spot where you download results, there's now an extra option, which includes the tracking part. So tracking needs to be run after the analysis job has completed. Now for algorithms that support tracking, there will be the CSV results will include the tracking information. Currently it's 3 buttons, it will be 2 buttons soon. So another thing we're running in parallel is the Arise Challenge platform. This is a test. We've invited student teams to join this effort. I think right now there's 4 or 5 active student teams from different countries that are working on the OPSIS challenge. So we have published the OPSIS training data and invited teams to develop better algorithms than the current one. So on the next slide, you see there's a lot of activity going on. People are submitting algorithms. So this is the preliminary leaderboard. And the next slide, you see the actual classification leaderboard. So currently Chirac is in lead, and the other teams we have to motivate to submit their algorithms. So hopefully next town hall, we actually have the final result of the AI challenge for the OPSIS.
Thanks a lot,
That was
Jaak.
it.
Yeah,
you're welcome.
And before we go to Lotte, so just a quick summary, right, of all that is happening. So you see that, we can ingest data from various sensors already. What we are still kind of putting the wrapper on is that authorization before we can actually allow a lot of people starting to use this platform, but most of those components are ready. You can also see that we won't have algorithms for every type of data yet, but that's work in progress that we're working towards. And the last bit that Jacob showed is that we've, in the meantime, in parallel, actually, also been developing this platform to improve or even build new algorithms. So all of those components together, of course, create this amazing end-to-end workflow. So we're going to shift to DNA. And Lotte, your microphone is horrible. There's a lot of background noise. And we can't actually hear you. So maybe try logging in again or sound by phone. Did I say that correctly, Jacob? Yep, I see. Yep.
Yeah.
Great.
I tell mute.
So, do you have any feeling, for instance, if you look at the monitoring demonstration sites, you see 8 of those sensors. Okay, with the radar, of course, that's a separate stream. But do you foresee very quickly that we can basically start analyzing all of those sensors within Arise?
Yes. Also, if those audio moths we can analyze with bird information, right? If some of them have cricket information, that they can then be analyzed with the Cycad cricket detector. Of course, every algorithm has its limitations, right? But
Yeah. Great. Yep.
Hi, I'm back. Is this better or not?
Much better.
Much better.
Welcome
Okay,
back,
great.
Lotte. And I'm gonna hand over so that we're moving then
Yeah.
to the other workflow of Arise from sample to DNA sequence.
Yeah, so we go from sample to DNA barcode. The sampling team and the sequencing team are working on the Dutch DNA species reference database. Let's have a look at all the steps that we take from collecting samples to publishing the results. So collecting, let's start with that. Next slide. We work together. Of course, we cannot collect all Dutch species with the little team that we have. So we work together with experts like Peter van Bracht, that talked before, and a lot of others that actively collect for us. And of course, they need to know what is still missing. So that's why we built this target list. It's an online tool where all the users can search on any taxonomic level. And it's been there for a while, but we did an update of it to make it more clear what is still needed as a species. So before we worked with percentages. So you had all these columns with percentages from 0 to 100, but a lot of users mentioned that it was not really clear what that meant. So now we added a new column at the one, over there, species status. And there is just stated it's wanted or unwanted. So it's way clearer what we still need. And we also calculate that in a different way. Before we just said if we have 3 specimens of species, then we have enough. But now on the right, you see also a new column collected. And there we list all the specimens that we collected, but that are not processed yet. So we don't have any barcodes yet. But of course, if we have many specimens collected already, we also might have enough. So now when we have 3 or more collected specimens or barcodes, the species becomes unwanted. So that's for the target list. Okay, so then you can collect samples for us. We have now 3 specimen kits, 3 different ones, because we want to make collecting, of course, as easy as possible. You see this huge new plant sampling kit. It's really 1/2 a meter big. That's almost finished, and all those kits contain everything you need to start collecting for rice. The specimen sampling kits, we actually are creating new ones because we already handed out 100 of them, and it's really working well for us. So we ran out of them, so we need to make new ones. For the plants and the mushrooms, we are going to work with smaller work groups of experts that will collect the specimens that we still miss for us. And for the metadata, we have the online tool ADA. Some of you know it already. So next slide will give you a little bit more information on that. It's a nice standardized method to collect metadata about the samples that we receive, so that we can also automatically process everything. Now you can register animal, plant, and trap registrations in there. Fungi will probably follow. And we have currently 123 users. What's new is that you can upload up to 5 images now. Before it was 1. And especially for plants, that's really important because you want to take pictures of the habitat, but also close-ups. And next thing that we will add is an eDNA registration button. That's mainly for the big new project of Naturalis Identity that will fully focus on eDNA, and that will also make use of ADA. Yeah, this I already mentioned. 100 sampling kits handed out. Busy creating new ones. People already waiting for them, I know. So just to give you an idea of the numbers that we are talking about, since the start of Arise, 91 collectors actually collected for us, and they provided around 60,000 identified specimens. So that's really identified specimens. But of course, there are also, as Peter also mentioned, there are a lot of species that we don't know about, the unknown biodiversity. And that's where our bigger trap projects come in. So we are now, since April, running a project in Lenebos together with Staatsbosbeheer. Lenebos is in North Brabant, and we are testing our workflow there, and of course filling the target list with this unknown biodiversity. So it's also good for us to know, okay, how much comes out of this, and how much can we actually process as a team. So if you click, you see that we received 48 sample containers. They Staatsbosbeheer empties the traps that we put out. We set up Malaise traps, pitfall traps, and we collected leaf litter on 6 locations in the area. So now 48 sample containers received, and we processed only 15 of them, which is, you might think, okay, that's not so much, but if you click, already 10,000 specimens came out of those 15 containers, and we expect to receive 120 containers. So you can imagine the huge amount of specimens that come out. So if we receive all the specimens and metadata, we take pictures of everything that serve as digital vouchers. This is our Keyence camera that I already showed a couple of times before. That's fully now in production. It's taking photographs every day. It can take 96 images of these plates of individual specimens in 10 to 15 minutes. So this goes really fast. But we are also working on a new camera system. Next slide. Yeah, so this is what we call the grid camera. And that is meant to actually do the same, but then for insect drawers, so for pinned dry specimens, to also automate that process. It's not yet in production, but we are almost there. So that is, then we actually have, for every possible scenario, we have a lot of automated workflows here. Yeah, okay, next one. So a couple of events coming up in September. First, we have the DNA validation workshop given by Peter van Bracht. Here he already talked a bit about what DNA can reveal. So if you are interested in, okay, so I have now my DNA results, what can I do with that? How can I validate it? How can I analyze it? And you want to dive really deep into that area and that material, then this might be very interesting for you. And then 2 weeks later, we have Barcoding NL. That's a day full of workshops, of keynote speakers, all around barcoding. You can actually already register by clicking or scanning the QR code here on the slide, or you can always send us an email and we will invite you. So then we go to the sequencing team.
Right, thanks a lot, Lotte. And indeed, Barcoding NL is not just for Naturalis, right? Not even just for Arise, but basically putting on stage all of those people that are contributing to making barcoding a new way of doing biodiversity monitoring. So please feel welcome. Kevin, all yours.
Yeah, thank you. Well, Lotte just showed us how many specimens we just collected. So I figured I'll show how many DNA samples we already processed. It's about half. So we still have quite a bit of material to go. Luckily, we're in the process of hiring 3 new people for the lab team. So come September, we have a lot of extra hands in the lab to help process this huge amount of samples that is coming from the Leenderbos. If we extrapolate this, I expect more than 100,000 samples. So there's a lot of specimens that need to be processed. We've spent some time optimizing a bit of the protocol. So the insect protocol is now finished. And we're already working on publishing that as well. So that will be available for everyone, hopefully in the not too far future. And on the side, of course, we are also optimizing some of the other protocols because not all the samples we get are insects. The identity project was already mentioned, environmental samples. Of course, within Arise, we're also doing some environmental samples. And to prepare the workflow in the lab also for the amount of samples that identity wants to collect. And so we've been working a bit on the soil protocols. So for the people who've worked with soil extraction kits are really expensive. So they're almost €8 per sample to do a DNA extraction, and then you just have one sample. With some tweaking of the existing Arise protocols, we've managed to create a working soil protocol for about 10% of the price of the kit protocol, and we're lowering that price even more by playing a bit with the volumes. So once identity gets started, they can use a much cheaper protocol. The data was just delivered to us, so we need to do some analysis to make final conclusions, but results are looking pretty good so far. Of course, to get some grip on all the specimens that we are processing in the lab, we need to get some better insights into all the data that we're creating. And of course, eventually all this data will be in the core sequencing cloud. Chantal will mention a bit more about that later. And of course, eventually also on the Arise platform where everyone can log in and see the results. But in the meantime, it's also important for us that we have a good look on the results. So that's why, together with a student, we built what we've called now the nanopore sequencing dashboard, which gives us these nice overviews of, for example, the success rate of plates. And we have a nice overview of one of the plates that we've processed, where we can see if there is barcodes or not. which will help us make a bit more decisions on which samples to reprocess later on. But of course, in the next slide, there's also a different view. So this is an overview of all the different sequences that came out, with a nice download button, so that at least us internally, if there's questions about sequences, we can quickly find them and download them and analyze them. And in the next picture, there's like one of these detail pages where we can see all the amplicon details. So which primers have been used, what marker it is. So this is all the information that we are now capturing, and that will also provide to Sequence Cloud. But we needed a quick way to make this accessible for us, because we get a lot of questions about specimens and samples and sequences. And digging through Excel files is not our favorite pastime. Of course, this is just the sequences that are generated, and there will be, like Lotte say, a workshop on like hands-on validation. But of course, we also want to do some automated validation to filter out those things that we can easily say are contaminants, for example, bacterial contaminants, or the odd Homo sapiens that's in there every now and then. So we've been working on the workflow. So this is a schema that sort of summarizes that workflow. We've created a lot of these schemas, but now we decided this is going to be the way. So we'll start building next week, and hopefully we have this up and running as soon as possible, so that all the sequence data that we send out into the world is at least a bit filtered for common contaminants and things we can already identify. Of course, that's what we've been doing here at Naturalis, but as you know, Westerdijk is also working hard on sequencing a lot of fungal cultures. So this is a bit of an overview from a project with Dutch soil samples, where there's already almost 4,000 soil fungi strains identified. Some of them still have preliminary identifications, not the final identifications yet. There need to be some sequencing of extra markers to make sure that the identifications are correct. And from those, there's already more than 900 new taxa identified. So that's new species, either for the Netherlands or sometimes also for science. Of course, there are also a few still preliminary. This is an example of some of the new species that have been discovered and described and published in the Netherlands, with names that I'm not even going to try to pronounce. There's a link below for those people that are interested in reading on this. And it's always nice to see these wonderful plates with the pictures and the nice areas where these specimens have been collected, basically, in the soil samples. In the background, we're also still working hard on publishing all of the what we call legacy data. So of course we've been barcoding since 2010, and not all of this data has been properly published yet. So we're making a big push now to make all that data public, so that many people can also start using this kind of data. And last thing, of course, like I already mentioned, we're doing some meta barcoding. So the soil samples I already mentioned, the data was actually delivered yesterday. So now we can actually start doing the analysis there. Also last week, or this week's actually, the first test samples with bird poop have arrived. So we can start working on the protocols there and actually start using bird poops as a sort of a biodiversity monitoring tool. Instead of having to collect insects, we just let the birds collect the insects for us. And then, of course, we're going to do some things with the malaise traps as well, because now, up until now, we've been sorting malaise traps and sequencing every individual specimen. At one point, we will switch to just sequencing the malaise trap as a bulk sample, because, well, most specimens, hopefully, at one point, we have sequenced, so that we can just use metabarcoding to quickly identify those specimens that we still need. Yeah.
Thanks a lot, Kevin. Final bits. I know that we are now 1 hour in. There's just so much happening, but now we're wrapping it up by going back to all that data.
Yeah. All right. Thanks, Elaine. Can you hear me? Same room, you know how it goes with speakers and all that. All right, so I just talked a little bit about the BioCloud. Can you skip to the next slide as the brain of Arise? But when we started to develop all these different data pipelines, next slide, we kind of realized that for the sensor and media related information versus the DNA and sample related information, we really needed two very different data models. So we now have a BioCloud stream and a core sequence cloud stream, but they're very closely connected and being developed by the same team in the same system. So we can kind of use what we already built for the DIOPS pipeline, but then change it to use the ADA and the nanopore data sources. So as I just said, we use the same code base, but especially in the enriched layer in the data processing, they use two different data models. And I think the good news here is that besides the DIOPSIS pipeline, we actually now have the first implementation of the data coming from ADA and the data coming from the nanopore, so the DNA barcodes belonging to those samples in the enriched layer. That's a very big achievement. We're not in production yet, but this is a very first step to actually showcasing multiple data pipelines. Next for us is then create a curated layer of that data so that we can export the data for the DNA dashboard. I think that was already, I'm going to hand over, I think, to Lotte to talk a little bit about the DNA dashboard. That was a super quick core sequence.
Yeah, so of course, when you hand in specimens for Arise to sample and to sequence, you want to know what comes out of it. What is the DNA result? I think we started in May with sending out results by email. So it's kind of a manual process. Of course, we have all the metadata and we have all the lab data, so that needs to somehow be brought together into one thing. But yeah, sending out emails is probably not the best way. So that's why we are building this DNA dashboard, where you will find all the information about your own specimens, and also all the details that you provided us through ADA or other or through a sheet. So yeah, if you click, I think I have some numbers. So yeah, as I said, we started sending the results by email. We sent it out 79 results of projects of in total 203 projects that we have now. This also includes the bigger trap projects, so it's not all identified projects here, but so we are hopefully in a couple of months you won't receive any emails, but you can log into this dashboard and explore everything yourself.
Thanks so much, Lotte. And thanks everyone for your attention. I hope you could really see both kind of an update on, well, this is what we've been doing so far, but also really big changes over those past 3 months where you can see everything coming together, digital environments being created, so that our aim is before the end of the year, we really have that platform with various services and data really integrated at your disposal. So becoming the Marktplaats or the Spotify of biodiversity. So thanks for listening.